Hello everybody, I am Deepika from Learno Hub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. So students, now we are going to start with our chapter number 4 of class 10 biology, that is absorption by roots, the processes involved. So we are going to talk about what processes are involved while roots absorb certain things, and we know what do they absorb, right? So they absorb water and minerals. So this is what we are studying from our junior classes, right? So let us discuss in detail how this absorption takes place. So starting with the first topic, that is plant physiology. So what is plant physiology? So basically it is a branch of biology which deals with the life functions or the metabolism that is taking place inside the plants. So we are talking about the functioning of the cell, the functioning of an organ, the functioning of an organ system, or the functioning of an organism as a whole. Correct? So what we are doing, we are not talking about the external features, we are talking about how the metabolism, how the life processes are being taken place inside the plant. So what are those processes that helps a plant to sustain, that makes a plant a living cell, right? A living organism. Correct? So this is what is plant physiology, the branch of biology which deals with the life functions or the metabolism of the plants. Correct? So let us talk about in detail how all this happens. Especially we are talking about the absorption by roots in this particular chapter, and this topic, absorption by roots, come under plant physiology because we are talking about the life functions of the plant. Correct? Now starting with the next topic, that is absorption by roots. So we know that the roots of a plant has many functions, right? The very first function that we can say is these roots help a plant to fix in the soil, right? So they hold the soil tightly, and even it is like the rains, the winds, the storms, but these roots hold the soil so tightly that plants, they stay at their position, and in very few cases they uproot and they fall, right? So the main functions of the roots are water and mineral absorption. So they hold the soil firmly, and their main function is to absorb the water and minerals. So these water and minerals are very important. These are the life sustaining functions of the roots. Now when this water and minerals are absorbed, they are conducted to the stem, and from stem they are sent to the different parts, the branches, the leaves, and all the other parts where the water and minerals are required. So this is the main function of the roots, absorption of water and minerals, right? The first topic, the need of water and minerals for plants. So what is the need of water and minerals for the plant? So we know that cell has a protoplasm. What is this protoplasm? Protoplasm is the living content of the cell. So what does this protoplasm include? So I will write this term over here, the protoplasm, right? So protoplasm is the living content which includes cytoplasm. So there is a cytoplasm, plus there will be nucleus, and what else, there will be cell organelles. So all these are the part of the protoplasm. So protoplasm has lot of water, but except for this part of the cell, we need a plant needs water and minerals, right? So where there are four things where water is very much essential. So the very first thing is photosynthesis, right? So we know that in photosynthesis, what happens, carbon dioxide along with water in the presence of sunlight and chlorophyll, it forms sugar plus oxygen gas is evolved. So what is happening? These four raw materials, they are coming together and they are producing what? Sugar, or you can say food, and oxygen gas is released, right? So even if one of them is absent, say sunlight is not there, or the chlorophyll is not there, or carbon dioxide missing, and even if the water is missing, so this reaction will not move further, right? So water is very much essential for photosynthesis. So the next is transpiration. So what is this transpiration? Transpiration is the loss of water vapors from the surface of the leaves, right? So what happens when there is a lot of hot conditions, like the weather is too hot, so in that condition, plant roots, they absorb water, and just for the cooling purpose, right? So this water is a cooling agent, right? So we have studied it in many different subjects, like when we talk about any industries also, so water is a cooling agent. So what happens? Plant roots will absorb water, and these will be released from the leaves in the form of water vapors, and this gives a cooling effect to the plants, right? So they are able to sustain themselves in the hot weather, correct? So then next is transportation. Now there are minerals which are being absorbed along with the water, and do you think these mineral ions will be able to move themselves in a plant? Like they are absorbed in by the roots, right? And they will move without anything or on their own to the different parts of the plant? So the answer is no. So what is the medium which is actually transporting all these materials? So water is the medium. So they are absorbed along with the water, minerals are absorbed along with the water, and they move to the different parts along with the water, right? In the same way, we will study this in higher classes, but when this food or the sugar is prepared by the leaves, so this sugar or the food that is prepared during the photosynthesis, that is also moved to different part, transported to different parts of the plant along by the water only, right? So to move ions, to move the minerals, to move transport water, sorry, not water but food, all of these transportations require water as a medium, correct? So here water is the medium, right? Water is medium for transportation, correct? So simple. And what is happening over here? Water is the cooling agent, right? Water is cooling agent, correct? Okay. So the next is mechanical stiffness. So now when water is present inside the plants, it provides turgidity. So we will study about this term in in further slides, right? So turgidity is possible only when a a plant has enough water. So this makes the plant cells stiff, and these cells or the plant, they seem to be swollen, and this provides stiffness to the plants, correct? So these are the four situations where water is very much needed by the plants, correct? Now the characteristics of roots for absorbing water, right? So we know that roots absorb water. Now what are the characteristics which provides this ability to roots to absorb more and more water? So there are three characteristics. So the first one is huge surface area of roots. So the roots have huge surface area, right? So we will be discussing each of them in detail in the coming slides. So let us first just try to know what are those three important characteristics. So first is the surface area of the roots, right? Second one is the root hairs. They contain cell sap which has higher concentration than the surrounding water, right? So these root hair, they have cell sap, right? Now the concentration of solute inside this cell sap is more as compared to the surrounding water, right? So what is this actually? So we will study this point in detail as well, then only you will be able to understand. Then comes the third one. Root hairs have thin walls. Now because they have thin walls, so the diffusion is very easy, right? So let us discuss each of this characteristic in detail. So then now the first characteristic, that is huge surface area of roots. So there was a botanist named H.J. Dittmer. So in 1937, he studied 4-month-old rye plant. So when he uprooted that plant, he found out that uh, there were lot of you can say the thread-like structures in the roots. They are called as root hair, right? Right. So it was something like this. So when he uprooted that plant, so he found out the root system to be something like this. So there were small thread-like structures, and when he laid these root hairs end to end, so he got to know that the root length of this 4-month-old rye plant was about 600 kilometers. So there were billions and billions. If you this was just a 4-month-old rye plant, but if you take any of other plants, so there are around billions of root hairs, and which have like a huge length, like many kilometers long. So this is what provides huge surface area, right? So the roots provide huge surface area, and when the surface area is huge, they have a lot of space to absorb water. So this is what happened. uh, this is what happens with the roots, right? So if you see this is a balsam plant, right? So the balsam plant is very common in our gardens, right? So if you see this balsam plant, if you try to uproot it, gently uproot it, you will see this kind of a structure. This is what you will be able to see. So this is the main root, and there are many root hairs, right? So these individually, these root hairs help in the absorption of water and minerals from the soil, correct? So the root hairs, they provide huge surface area which facilitate the absorption of water. So the more the surface area is, more is the space to absorb water, correct? So this is what the huge surface area of roots. I hope this is clear. So how the surface area is increasing? This surface area is increasing because of the tiny root hairs that are present along the roots, correct? Now the second characteristic, that is root hair contains cell sap of a higher concentration than that of the surrounding water. So now we know that a plant root has millions and billions of root hairs, right? So each of these root hairs, so what are they? They are the extension of the outer cells of the roots, which have large vacuole. So they basically have large vacuole which are filled with the cell sap, right? And they have dissolved salts in them, correct? So now see, if we talk about a root hair, see, if this is a root hair and this will be the surrounding water, right? So now this root hair has these extensions of the outer cells of the roots. Now they have the large vacuole. So if I see, this is the cell of the root hair, so it will have a large vacuole, and in this large vacuole there will be cell sap, and this cell sap will have some dissolved salts, right? So what do they have? Dissolved salts. And if we see the surrounding water, so this surrounding water is just water, and the concentration of the solute is less. So this is the concept that we are going to discuss in detail in this chapter. So when we are talking about the concentration, so we are saying higher concentration of the cell sap, so this means we are talking about the solute. This means this cell sap has more solute in them. So what is this solute? This solute is, say, ions or these dissolved salts. So these are the solutes, and the water content is less, right? So when we compare this cell sap with the surrounding water, so surrounding water has more of the water molecules and less of these dissolved minerals, right? No, now the concentration of the dissolved minerals is more in the cell sap and less in the surrounding water. So what will happen? There will be movement of water from outside to inside of the cell, right? So water molecules are less inside the cell sap, right? So the water from the surroundings will move into the root hair. So this is how these cells, these root hairs are able to pick water, absorb water and send it to different parts of the plant. Correct? Now the third characteristic, that is root hairs have thin walls, right? So this is the diagram of a root hair, or you can say the cells in the cells of the root hair, right? So like every plant cell, so we know that every plant cell has a cell wall and a cell membrane, right? So there are two coverings basically, but in case of animal cell there is only one, right? So there is only cell membrane and cell wall is absent in case of animal cells. So in case of plant cells there are two layers, cell wall and the cell membrane. And this cell wall is, talking about the cell wall, so this cell wall is thin and it is permeable. So if you see this structure, so this is the epidermal cell, as you can see. So what is it? It is an epidermal cell, right? So these all are epidermal cells over here. From starting from here, and all these are epidermal cells, right? And now in this particular cell, so this has a fully grown root hair is shown in of this particular epidermal cell. So outside here is the cell wall, inside of the cell wall is the cell membrane, and between is this content is the cytoplasm, and these large structures are the vacuoles, right? This small structure is the nucleus, and this is the fully grown epidermal hair, right, or the root hair. Now if you see this structure, this cell, this is also an epidermal cell, but here the root hair is just beginning to grow, right? So the root hair is not fully grown, it is like in the initial stages of growth, and same is with this cell, right? So basically what happens, there are two walls. Now when the content has to move from outside to inside the cell, or vice versa, from inside to outside, so all these content, even if it is water, it is salt, it is any kind of ion, so it has to, what it has to go through or face both of these membranes, right, or these layers. So in case of cell wall, this cell wall is thin and it is permeable, it is freely permeable, so it allows all the type of the content to move throughout it, right? So water molecules will also move, then dissolved molecules will also move from in and out of the cell, right? So the next layer over here is the cell membrane. Now the cell membrane is again thin, right? So this cell membrane is thin, but it is semi-permeable, right? So we said that cell wall is permeable, but cell membrane is semi-permeable. This means it will allow the water molecules to move, to pass through it, but it will not allow the larger molecules to pass through it. So basically there is some restriction. It is not allowing all the content from outside to pass into the cell, right? So this cell membrane has the major role to play in case of absorption of water, right? So I hope all the three characteristics are clear to you. Let us recall them again. So first is the large surface area of root. So if you lay down these root hairs, there are billions of root hairs, and if you lay down these root hairs end to end, they will it will take many kilometers to completely line them up, right? Second one is they have the cell sap. Now this cell sap has more of the dissolved salts, and outside medium, outside water, the surrounding water has less of these dissolved salts. So it is the tendency of the cell, or it is the tendency of water to move from the lower concentration to the from the higher concentration to the lower concentration. So there is higher concentration of water molecules outside the cell and lower concentration of water molecules inside the cell. So water in the surrounding of the root hair will move into the cell, and hence this is how water is being absorbed by the cell. So this was the second characteristic. Now the third characteristic, root hairs have thin walls. See, if anything has thick walls, so that will actually restrict the movement, right? When the walls are thin, so the movement will be easy. So in every plant cell there are two layers, right? So one is the cell wall, one is the cell membrane. Now this cell wall is very thin and it is freely permeable. It allows all kind of material to pass through it. But then comes the cell membrane, which is thin but it is semi-permeable. So it allows the water molecules to move, but it does not allow the large molecules to pass through it. Right? So this cell membrane restricts the movement of some of the content from outside to inside the cell, and this creates a difference in the a difference in the characteristic. So basically this is the secret of absorption of water, right? So I hope these 3 points are clear to everyone. Now the next topic is absorption and conduction of water and minerals. So just now we studied that there are some important characteristics of roots which help in the absorption of water, and now is the time to discuss the absorption and conduction of water and minerals. So basically these roots have certain characteristics which help in the absorption of water, but there are certain phenomena which actually are taking place and help in the absorption and the conduction of water from roots to different parts of the plant. So basically there are 5, right? So the first one is imbibition, second is diffusion, third is osmosis, fourth is active transport, fifth one is turgidity and flaccidity, right? So it is not at all difficult to understand them, right? So you can always make a mnemonic out of it. So it is I D O A T F, right? So I thought it will be I for I, right? I do not. So like is just to add to the line, right? I do not like onion and turmeric, right? So I do not like onion and turmeric finally, right? So it is like I tasted something, I did not like onion and turmeric taste, right? So this is my way of recalling these phenomena, right? So you can always make your own mnemonics, correct? So now we will be discussing each of these phenomena in detail. Now talking about the first phenomena, that is imbibition. So talking about imbibition, what is imbibition? Basically it is a phenomena in which a living cell or a dead cell tend to absorb water from its surroundings. So there is a phenomena of surface attraction. So these living or dead cells, they have ability or they have this property to attract water from the surroundings. So there is surface attraction, right? So in this case what happens when there is any substance which is made up of cellulose or protein, so these substances are hydrophilic. So this word hydro will break into two, one is hydro and another is philic, right? So hydro means water and philic means loving, right? So there is attraction to water, so they have the strong affinity, right, to water. So now these substances which are made up of cellulose or proteins, they attract water from their surroundings. Say if there is water in the surroundings or there is a moisture in the air, so they attract this. This water get attracted to these substances, and in turn these substances they swell with water, or they imbibe water, they swallow water or they imbibe water, and finally they swell up, right? So they their structure or their body, they swell up because of this absorption of lot of water. So now what happens due to this, we have seen whenever you remember, you want to eat those sprouts, so what do? You put the seeds in the water, and then you see those seeds, they get ruptured, right? So they take up lot of water, they swallow lot of water, and their seed coats, they start to rupture, right? And if you now take them out from the water, put them in a moist cloth, keep them in a dark place, then they tend then you see something like this, right? So the wall, the coat, the seed coat is ruptured, and these structures, these extensions start coming out from the seed, right? So basically what happened, the seed coat gets ruptured in case of the germinating seed. When the seed is germinating, the seed coat gets ruptured, right? So how this happens? This all happens because of the process or the phenomena called imbibition, right? And this is very important phenomena for the ascent of sap, right? So we know that what is ascent of sap. So the absorption of water by the roots and sending it to all parts of the body, right? So sending them in the upward direction, that is the ascent of sap. So imbibition is one of those important phenomena which help the plants to absorb water, correct? Now talking about the second phenomena, that is diffusion. So what is diffusion? So diffusion is basically the movement, the free movement of molecules from the region of higher concentration to the region of lower concentration, right? So we will now just discuss the definition of diffusion. So it is the free movement of molecules of a substance from the region of their higher concentration to the region of their lower concentration when two are in direct contact. So diffusion will only happen when the two surfaces are in direct contact with each other, right? So say if this is a root here, right, and there is lot of water in the surroundings, and inside these cells the water is in less quantity, it is in lower concentration, right? So the water molecule will move from outside to inside. So how this is happening? When this water and this cell is in direct, this root is in direct contact with each other, correct? So now let us understand this phenomena of diffusion with a simple experiment. So in this experiment, what we have done, we have taken a beaker, and inside the beaker we can put say sugar, we can put a little amount of salt, or we can use a soluble dye, or we can use a potassium permanganate, right? So the diffusion of a soluble dye, potassium permanganate, or you can use a sugar cube, or if there is a salt, right? So you can use any of them, right? So now what will happen? So this particular experiment will progress something like this. If you see the A beaker, basically this is the same beaker throughout, but at the stage A or the stage 1 you can say, so we have put this potassium permanganate over here, and now what has happened? So you see the water over here is clear, right? So there is no solute over here, but more of this color, right? So why we are using this soluble dye or the potassium permanganate? So the situation becomes more clear when we see the color difference, right? So you see the solute is more concentrated near this tablet, right? But when the time passes, as the time progresses, the experiment also progresses. Now you can see that these solute particles, they are diffused to the other parts of the liquid where the solute concentration was less, correct? Now when we come on to the stage 3 or the C stage, then we see that there is uniform distribution of these solute particles throughout the solution. So this is what happens with the diffusion. So this is the first stage, this is stage 2, and this is stage 3. So initially these solute particles, they were concentrated at a one position. Then these solute particles started to move to the other positions, to a position where the concentration of these solute particles were less, and finally this solute is completely dissolved in the solution, and now this liquid has become homogeneous. This particular solution has become homogeneous solution, right, where the concentration of solute and the solvent is equal throughout the solution, correct? So this is a simple experiment of diffusion which you can also do at your home using any soluble dyes, right, or the sugar cubes, correct? So just to make it more like homogeneous, you can stir it with the spoon or you can stir it with the stirrer also, correct? Now the third phenomena is osmosis. So now looking at the osmosis and the diffusion, so the definitions are somewhere the same, right? But not the same. Now, if you talk about osmosis, so what is osmosis? So let us go back to diffusion first. So what was diffusion? So in diffusion, we saw there is free movement of molecules of a substance. And what was that substance? That was basically the solute, right? So we did that experiment, right, where the solute particles were moving to the locations where the solute was less, right, and there was free movement. There was no barrier in between, there was no obstacle in between. But in case of osmosis, there is a little twist. So again, there was the movement from higher concentration to the lower concentration in case of diffusion. The higher concentration of the solute to the lower concentration of the solutes, right? So this is what we experimented in the last slide, right? So those little dye, soluble dye or the potassium permanganate was restricted to one part of the beaker, but these solute particles, they spread throughout the beaker and they made it a homogeneous mixture. But when we come to osmosis, there is movement of water molecules, there was movement of solute particles, now solute molecules, and now here there is movement of water molecules, but the direction remains the same, from the region of their higher concentration to the region of their lower concentration through the semi-permeable membrane, right? So what is happening? So there is movement of water molecules now from the region of higher concentration to their region of lower concentration, and now there is a barrier or there is an obstacle course in between, and that is the semi-permeable membrane. So remember, in case of diffusion, the solute particles were moving from their region of higher concentration to the region of the lower concentration. But in case of osmosis, there is movement of water molecules, correct, from their region of higher concentration to the region of lower concentration, and in between them there is a barrier, which is what? That is a semi-permeable membrane. So a small difference, and I know you are now very well aware of the difference between osmosis and diffusion. So there are two more terms that are associated with osmosis. So it is like from where to where this movement is taking place, from inside the cell to outside the cell, or from outside the cell to inside the cell, right? So let us make a cell. So say this is the cell, this is the surrounding medium, right? So we take it as the surrounding medium, correct? Now when there is inward diffusion, right, so what is happening? the water concentration is higher outside the cell than inside the cell, right? Because there is movement of water molecules from their higher concentration to their lower concentration. Now in this case we say that water concentration is higher outside the cell. So what will happen? The water molecules will move from outside the cell to inside the cell, and this condition is called as endosmosis, right? Now there is another condition, right? So this was the cell. Now we'll take another condition. Now this is the cell. Again, this will be the surrounding medium. And now in this case, the water concentration is higher in the cell, right, than outside the cell. So in this case, water molecules will move out of the cell into the surrounding medium, and this is called as exosmosis, right? So what do we call it? We call it as exoosmosis. Now in this case, when the water molecules are coming into the cell, so what will they do? They will actually make the cell turgid, the cell will swell up. But when the water molecules are going from inside to outside the cell, so now the turgidity will be less and the cell will shrink now, right? So if there is a bag, right, so if there is a handbag, I'll take the example of my handbag. So if I have a handbag and I put a lot of stuff in it, what will happen? That handbag will actually swell up, right? Now I came back home. That time I was in the some shopping mall, so I was buying things and I was stuffing those things inside my handbag, right? Now I came back home, I emptied my bag, right? I took out the stuff from my bag. So what will happen to my bag? My bag will shrink. The same is happening in this case, in case of endoosmosis and exosmosis, right? So in endoosmosis, water molecules are coming into the cell, so the cell is swelling up. In exosmosis, water molecules are more inside the cell, so they are going to outside the cell, right? So in that case, cell will shrink. Correct? So I hope this is clear. Right. So now we will be discussing osmosis experiment. So how are we going to do this osmosis experiment? So it is super duper interesting and super duper easy, right? So this is our experimental setup. So what is there in the experimental setup? I will explain it to you. So this is what? This is experimental setup, right? So in this experimental setup, we have taken this clamp stand, right? And we have taken a thistle funnel. So thistle funnel looks something like this, right? So it has a wide mouth, open mouth, and a long tube over here, right? So what we will do, we will put a sugar solution inside this thistle funnel. So there is sugar solution. Now we will cover this mouth with the cellophane sheet, right? And now we will invert this thistle funnel and put it into the beaker containing water, right? So as soon as we put this thistle funnel inverted into the beaker filled with water, we will mark the initial level of this sugar solution, and this is very, very important. Correct? So if we do not mark this initial level, how will we come to know whether the osmosis has taken place or not? Correct? So this is our experimental setup. Now when we are carrying out an experiment, we also need one control setup, right? So this is control setup. So what we are going to do in the control setup, so it will exactly look like this, but the difference lies in the material or the solution which is there inside the thistle funnel. So inside the thistle funnel in the control setup, there will be water, same water that we are putting in the beaker. Correct? So that will be a control setup. So now what will happen after some time, or you can say after few hours, when we will notice both the setup, so we will see that in case of thistle funnel, the level of this sugar solution has increased. So this was the initial level, and now the final level is going to be somewhere like this. So level rises basically. Correct? So now In case of control setup, what will happen? There will be no change in the level of the solution inside the thistle funnel. Correct? So this is what happened inside the thistle funnel. Now talking about the water content in the beaker. So in case of experimental setup, this water level will come down. So initially the water level was here. So this was the initial level and it will slightly drop, and this will be the final level of the water inside the beaker. But in case of control setup, it is going to be the same, right? The water level, the initial and the final levels inside the beaker are again going to be the same. Correct? So these are some of the observations that we found from this osmosis experiments. So based on these observations, we will make certain conclusions. And there was one more observation. Try to taste the solution, the sugar solution, and then try to taste the water in the beaker. So you will find that water inside the beaker in the experimental setup was not sweet. Right? So now let us look into the conclusions that are made on the basis of these observations in this experimental setup. So now let us look at the conclusions. The first conclusion is water has passed through the cellophane sheet with which we covered the thistle funnel, correct? To enter the thistle funnel containing the sugar solution. Now we saw that there was a rise in the level of the sugar solution, correct? But in case of control setup, there was no change in the level of the solution inside the thistle funnel, and there was slight drop in case of the water level in the beaker. Correct? So what did this show? That the water has entered from the beaker into the thistle funnel through this cellophane sheet. Right? So water has entered into the sugar solution, due to which the level has increased. The level of the sugar solution inside the funnel has increased. Correct? Second is sugar from thistle funnel has been passed into the beaker. How do we know that? We know that because when we tasted the water in the beaker, it was not sweet. Since sugar solution is going to be sweet, right? So since the sugar solution has not passed from the funnel into the beaker, so that is why the water in the beaker is not sweet. So this means sugar molecules have not passed from funnel to the beaker, right? So this is our second observation. Now coming on to the third one. Cellophane paper act as a selective or differentially permeable membrane. So now this cellophane sheet has played a very important role. It is a selectively permeable membrane. So it allowed only water molecules to pass from beaker into the funnel, but this membrane did not allow sugar molecules to pass from funnel into the beaker, right? So this acted as a semi-permeable or differentially permeable membrane, correct? So I hope these conclusions are clear. Now let us modify this osmosis experiment. Now this is modified experiment where in case of the beaker water, what we will put? We will put less concentrated sugar solution, right? So what we are going to do is we are going to put less concentrated sugar solution. But in case of the first experiment, the osmosis experiment that we carried out, we have taken water in the beaker, right? So what will be the observations? What are the observations going to be in this modified experiment? So they are very simple, right? So again there is a cellophane sheet which is a selectively or differentially permeable membrane. So this membrane will only allow water molecules to pass through it and not the sugar molecules, right? So what will happen? Where is more amount of water? Because in osmosis, what is moving? Water is moving from its higher concentration to its lower concentration. So water molecules in the beaker are more as compared to the water molecules inside the funnel, right? So what will happen? Now the water molecules will move into the funnel from the beaker, right? So again the level is going to rise in the thistle funnel, and there will be slight drop in the level of the solution inside the beaker. Simple? Clear? Let's see some more modifications to this experiment. Now let us modify this experiment, this osmosis experiment a bit more, right? So what we will do now, so in the previous one, what we did, we did exactly the same, just we changed the solution of the beaker. Now what we are going to experiment is with the sheet. So we were using cellophane paper over there, and now what we are going to do is we are going to change that membrane, right? So first of all, we will be taking a rubber sheet. So in case of a rubber sheet, what will happen? So in case of a rubber sheet, there will be no change in the level of the solution. Now since it is a rubber sheet, so it will not allow even a water molecule or the sugar molecules to pass through it, right? Because it is an impermeable membrane which do not allow any movement. So no movement of any molecule, right? So what we will observe that there is no change in the level of sugar solution, and since and in the same way no change in the level of water in the beaker, right? Now the next that we are going to take is muslin cloth. Now this muslin cloth, we know it has large pores, right? So if you cover the beaker, if you cover the mouth of the thistle funnel with this muslin cloth, it has pores, it has large pores which cannot hold back even the sugar molecules. So water will move freely, right? So the water molecules will move freely, and also there will be movement of the sugar molecules. Now because of gravity, now there are sugar molecules and this thistle funnel is inverted. Now due to gravity, these sugar molecules will also come down, and they will get dissolved with the water in the beaker. They will come down from the thistle funnel into the beaker containing water, right? So this shows that this muslin cloth is a freely permeable membrane. So this allows even the larger molecules to pass through it, and hence there will be complete mixing of the solutions in the funnel and in the beaker, right? Now again a small twist in the experiment. Now, the another modification in the experiment is with the whisking bag, right? So, what is this whisking bag? So, this is a bag, something like this, and what we will do, we will put some sugar solution into this whisking bag, and now we will insert a capillary tube into this bag. Now, we will wait till some of the sugar solution rises into this capillary tube, and then we will tie this bag with this tube tightly, right? Now we will do the same thing. We will put this bag into a beaker containing water, and now what we will happen, that we have to see. So after some time we will observe, after few hours say, we will observe that there is certain rise in level of the sugar solution in the capillary tube. Now why this happened? Because the water molecules from the beaker has entered into this bag, and due to which the sugar level has it has increased in the capillary tube, right? So what is happening again? Again the water molecules are moving from its higher concentration to the lower concentration, from the beaker into the whisking bag, and since there is only movement of water molecules, so this means that this whisking bag membrane, it also acts as a semi permeable membrane which allows only water molecules to pass through it. And do you know where is this whisking bag used for? It is used as a dialysis bag, right? So in case of patients where they have their kidney dysfunctions or kidney failures, right? So in the dialysing unit, these whisking bags are being used, right? So this is how we got to know that how osmosis take place. So in case of osmosis, there is just the movement of water molecules from the region of its higher concentration to the region of its lower concentration and through the semi-permeable membrane. So out of all of them, how many membranes we used? We used four, right? The cellophane sheet, then the rubber sheet, then the muslin cloth, and lastly the whisking bag. So out of them, we found out that only the cellophane sheet and the whisking bag, they acted as semi-permeable membranes, which allowed the water molecules to pass into the sugar solution. And in rest of the cases, in rubber sheet, there was no movement at all because it was an impermeable membrane, and the muslin sheath had large pores due to which it was freely permeable membrane, right? So this is how osmosis takes place, right? So presence of a semi permeable membrane has to be there when the osmosis is taking place, right? Semi permeable membrane, selectively permeable membrane, membrane which allows only water molecules to pass through it and does not allow any other molecule, the large molecules, to pass through it. Correct? So from the above experiments, the four different experiments of osmosis, we have observed, we have concluded these 2 points, that while we are carrying out osmosis experiment, we need two different concentrations of liquids, right? So say one is a sugar solution, another is water. So how we can refer to them? So the two liquids of different concentration. So we use the term tonicity for the two liquids of different concentrations, right? And now how can we refer to them? So either we can refer the tonicity in terms of the water molecules. So one has higher concentration of water molecules and another has higher concentration of lower concentration of water molecules. Or so the first is in terms of water molecules, right? Now the second is solute molecules. So in second, we can say, in the second way, we can say that this particular liquid has more concentration of sugar solution or the solute, and this particular liquid has a lower concentration of the solute particles, right? So this is the way how we refer to liquids, their different concentrations, two liquids of different concentrations, right? And the second thing that we concluded is that when these two liquids, they are separated by a cellophane sheet or the whisking bag, right? So it behaved as a semi permeable membrane, a membrane which allowed only water molecules to pass through it and does not allow the larger molecules to pass through it. And from where this movement was taking place, so the water molecules were moving from their region of higher concentration, the region where they were in more concentration, to the region of the lower concentration, the region where they were in the lesser concentration. Correct? So I hope this is clear. So now the question is, how long can osmosis continue? Now the osmosis will continue till the solutions on both the sides are on the same level, right? So the concentration is same on both the sides of the solution, right? Is it so? But it is not true in all the cases. So what will happen, we will study with the help of a diagram. So this is a semi permeable membrane, and this is solution A, this is solution B. So in the solution A, it is more diluted one where the water molecules are more. So these round ones show the solute particles, right? So this is a solute particle and these smaller ones are water molecules. Correct? Now in the solution B, these solute particles, as you can see, are more as compared to the water molecules. So basically the concentration of solute particles is more on the side B, right? So what will happen? There are less water molecules. So now the water molecules will go from solution A to solution B, from this side to this side. Correct? Now since the pores are not large enough so that these what do we call them, these solute particles, they cannot move, Since this is a semi-permeable membrane, so it will allow only water molecules to pass through, not the solute molecules, right? So now if the pores were large enough, sugar molecules would have passed to the another side, right? So this membrane, in that case, this membrane has to be a freely permeable membrane, right? A permeable membrane, but this is a semi-permeable membrane, so water molecules will only pass. Now just recall that experiment where we were using the thistle funnel, where there was a sugar solution and it was kept in a beaker containing water. Now this osmosis will continue, right? The water molecules will continue to move from the beaker to the thistle funnel, but a time will come when there will be no osmosis, because the height and the weight of the column is increasing as the water molecules are going from beaker to the funnel, but there is no movement from funnel to the beaker side, correct? So the funnel's weight and height are, column height are being increasing. Now a time will come when the pressure will be put from the other side, from the top of the funnel, to stop this movement. So that pressure will be enough to stop the process of osmosis, and now no further movement of water molecule will occur from beaker into the funnel, right? So there are certain barriers, there are certain stages, there are certain restrictions which stop this process of osmosis, correct? So I hope this is clear. Now coming on to the next very important term, that is osmotic pressure, right? So in the previous slide we were talking about how long this osmosis will continue. Will it continue for long um, till the concentration on the both sides is not equal? But the answer is no, and in many cases the osmosis might not happen even though there is concentration difference, even though there is a semi permeable membrane that is present. So what is the reason? So the same setup that we were using for osmosis, right? But there is a little twist in this setup. Now we have put a piston with the weight. So there is a piston with the weight. Now this was the initial level, and over here we have put a piston which has a certain weight, and due to which there is a lot of pressure that is being applied from the top and which nullifies the osmosis, correct? So what will be the definition for osmosis? So it is basically the minimum pressure. So what we are doing with this piston, we are putting pressure, right? So it is basically the minimum pressure which is exerted to prevent the passage of pure solvent into the solution when two are separated by a semi-permeable membrane. So it is basically the minimum pressure, right? Or we can also define it something like this: the measure of its tendency to take in water by osmosis, right? So what we are doing, we are putting a pressure, and due to which even though these two solutions, they are separated by a semi permeable membrane, but there is no movement of water from its high concentration to its lower concentration, right? So this is osmotic pressure. Now talking about the term tonicity. So tonicity is the term which we are now using for a while. So what is this tonicity? Tonicity is basically the concentration of the solutions that determine the direction and the extent of diffusion. So in which direction the diffusion is taking place and to which extent the diffusion will take place. So basically this tonicity has three important terms. The first one is isotonic, right? So the word iso means same, and tonic means it has been taken from the word tonus, right? From the word tonus, which means tension, concentration, right? So the same concentration. So we are assuming a cell is placed in a solution where the solution outside the cell and solution inside the cell both have the same concentration. So in that case there will be no net movement of water molecules, like, so there is no net movement. Okay, so in this case there will be no osmosis. Correct? Now, the second term is hypotonic. Hypo, the word hypo means less, right? Tonic means the tonic concentration, or just say the concentration. So, less concentration of what? So, we are talking about the less concentration of solute. So, now again we are assuming the cell, and a cell is present in a solution which has less concentration or the lower concentration of solute. So which means the concentration of the solute is less in the solution, but inside the cell the concentration of solute is more, right? So the solution outside the cell has lower solute concentration and than the fluid inside the cell. So in this case, what will happen? This means the water molecules are more outside the cell and water molecules are less inside the cell. So water will move from outside to inside the cell. So which means what is happening? Endoosmosis is happening, right? So what is happening? Endoosmosis. So what will happen? Cell will swell up because water molecules are moving into the cell, right? Now the third is hypertonic. Hyper means more, right? So more concentration. So it is the solution. It is a solution in which so there is a cell and it is present in a solution that has more concentration of solute. So the solute outside the cell are more. So now what will happen? From where will be the movement of water? So now the water will move from its region of higher concentration to lower concentration, from inside to outside the cell. So in that case, what will happen? Exoosmosis will happen. The water will move from inside to outside the cell, and in this case, cell will shrink. Correct? So simple. These three terms are very, very important. Please understand them and relate them with each other. We always use a diagram that we will be using in our next slide, right? So let us now move on to our next slide, where we have taken one animal cell and one plant cell, right? So this animal cell is going to be RBCs, right? The red blood cells. So we are going to perform the tonicity experiments on these cells, right? So with looking at the diagrams, looking at what is written along with the diagrams, we will find out in which condition or in which concentration the cell has been placed, right? So is it a isotonic solution? Is this a hypertonic solution, or is this a hypotonic solution, right? So the first one is in this case the RBCs are inside a solution where the water molecules are entering into the cell also and outside the cell also. So water entering the cell is equal to the water leaving the cell. So same is in this case. So this is a plant cell. So there is no net gain of loss, no net loss or gain of water by the cell. So what type of condition is this? So this is a isotonic solution, right? So this is a isotonic solution. Now the next comes is a cell when it is placed in a solution where water is leaving the cell, right? So water entering the cell is less as compared to the water leaving in the cell. So what is happening? Exoosmosis, which means water from the cell, inside the cell, is going to the surrounding medium. So what is happening now? The cell is shrinking. So in both the cases we can see the cell shrinking, right? So what kind of solution is this? This is a hypertonic solution, right? A solution where the water molecules inside the cell are more as compared to the water molecules outside the cell, right? So this is a hypertonic solution. Now comes the third one. So we know that now this is a hypotonic solution, but still we will discuss it a little bit. So this is a RBC, right? So what has happened now? There the water is entering into the cell. A lot of water is basically entering into the cell, leaving it like the cell is swelling, right? The cell is swallowing water, and now the cell has swollen up, due to which what has happened? endoosmosis has happened. Now the cell has taken up so much of water that now the cell has burst, right? And the same is happening in this case. Now the water has entered into the cell, which means the water in the surrounding medium is more as compared to the water in the cell, right? So endoosmosis has taken place. So what is it? This is hypotonic solution. The cell is placed in a hypotonic solution, right? So this is tonicity, correct? So let's move on to our next topic. Now, in case of plant cells, there is one more important factor that is actually helping out the cell, and that is the extra layer of wall, that is the cell wall. Cell membrane is present in both the cases, in both plant cell and in animal cell, but there is one more striking feature in case of plant cell, that is the cell wall, which is very rigid, the rigidity of the cell wall, which actually prevent it from bursting, correct? Now the next topic, that is active transport, the fourth phenomena, that is active transport. So what is this active transport? So it is basically a passage of substances from lower concentration to higher concentration. Till now, we were discussing how the molecules are moving from higher to lower concentrations, right? So say if water is at a higher concentration at some place, so it is moving to the other solution where the water is at lower concentration. Now the situation has come where these molecules, they are moving from a lower concentration to higher concentration, right? So through the living cell membrane, using some energy in the form of ATP, right? So energy is always in the form of ATP that plants and animals use. So what will happen now, this movement is against the gradient, correct? So we already have a lot of say lot of solute in or water in a solution, but we are still forcing some more water to get into that particular solution, right? So how is this possible? How we can force that solution? We are forcibly putting that solution, right, or those molecules into that solution. So how is it possible? It is only possible with the help of energy. We have to put a force. For that force, what do we need? We need energy in the form of ATP, correct? So this active transport is in the direction opposite to the diffusion. So diffusion, from where it happens? From higher concentration to lower concentration. But in case of active transport, the movement happens from lower concentration to the higher concentration, correct? So we are, that is what I was saying, we are forcibly pushing these ions into the solution, and for that we need energy. And now there is a passive transport, right? So it is actually opposite to the passive transport, correct? So basically we can say that the movement of molecules from higher to lower concentration, where we do not need to put any extra energy, is the passive transport, as diffusion, as osmosis, right? So those are the passive transports. And over here, where we need to put an extra energy to push the molecules from lower to higher concentration, is the active transport, correct? So why do we need this active transport? So basically there are certain ions, say the potassium ions, sodium ions, which are already there in the roots, right? But still plant need more. So we need to push them into the roots so that they can be transported to the different parts, right? To the different parts of the plant. So in that case, active transport is very much needed, right? So in that case, we need to forcibly push these ions into the root hair, putting an extra energy, correct? So I hope all the topics that we have covered in this video are crystal clear. Now the rest of the topics and few important questions we will take up in the next video. So till then, as you know, Learner Hub, free hair per best hair.